Hi everyone. Today we are going to continue our AMP1 course with Chapter 11 on the fundamentals of the nervous system and nervous tissue. Basically, the rest of the semester, the rest of our content is going to be on the nervous system. So we're going to do the fundamentals of the nervous system in general, the cells, the connections, things like that. Then we will do central nervous system in Chapter 12, peripheral nervous system in Chapter 13, and the autonomic nervous system in Chapter 14. So we're going to start out with the fundamentals of the nervous system today. So this video that you can find on Mastering talks about drugs and how they affect a patient's nervous system. So the functions of the nervous system in general is to be the master controlling and communicating system of the body. They're going to communicate through electrical and chemical signals that are rapid, specific, and immediate. The nervous system function can be simplified as a sensory input, any kind of gathered information, an integration center where decisions are made, processing, interpreting, and you'll see the picture of the brain there with the little green arrow. And then the motor output, which is some sort of effector organ response, a muscle contraction, a glandular secretion, something like that. This one is showing water, and the person obviously wants to pick up the glass of water, so you see it, you lift it, right, and you pick it up. Sensory input, integration, and motor output. The central nervous system is divided into two parts, sort of the integration part, which is the central nervous system, the brain and the spinal cord, in the dorsal body cavity. Then the input and output, the sensory input, motor output portion of the program, which is the peripheral nervous system. That are going to be the spinal nerves, the cranial nerves, right? Things that leave the central nervous system and go out to the body. So the peripheral nervous system has the two divisions, right? If we want to talk about the input, the integration, and the output, the peripheral nervous system is going to be the sensory, the afferent, the towards, right? Afferent means come towards. Division, somatic sensory fibers and visceral sensory fibers convey impulses towards the central nervous system. We're gathering information. So sensory, afferent, gathering, right? The A in afferent goes with gathering information. The motor or the efferent division transmits impulses to the effector organs, meaning to the muscles, to the glands. And we're going to do that voluntarily, like with our muscles. Right? If I want to pick up this can of seltzer, then I'm going to contract my bicep. I'm thinking about that. That's somatic nervous system. Motor division, so it's still output, but it's somatic, I'm thinking about it. The autonomic nervous system would be glandular. Like if I think of jelly beans or Sour Patch Kids, you get this happening, right? With the salivary glands. That's an autonomic nervous system, right? I didn't force my salivary glands to squeeze. They did that based on a thought that I had. So sensory input, motor output. And again, the somatic nervous system is voluntary. The autonomic nervous system is involuntary, something that the body will just do as a result of the regulation of smooth muscle, cardiac muscle, glands, things like that. With the autonomic nervous system, because there is no sort of on-off, right? If I want to contract my bicep, I can stop it anytime because I'm thinking about it. Right? There's an on and an off. When the autonomic, if you're not consciously thinking about it, we have two divisions. We have one that sort of raises, increases energy, and we have another that decreases, resting, digesting, right? Sympathetic goes up, resting and digesting, parasympathetic goes down. And we'll do that more in Chapter 14. So here it is in a little table. So you have the central nervous system on the left, you have the peripheral nervous system and all of its divisions on the right. You'll notice that the little green arrow goes from sensory, or is it blue? I can't really tell. A blue arrow or whatever goes from sensory up towards the central nervous system, and then the red goes down away. So the peripheral nervous system contains both sensory input fibers, motor output fibers. And then we have the division between the somatic voluntary, the autonomic involuntary, and from the autonomic, the sort of resting and digesting, and then the increase in energy, the sympathetic and parasympathetic. Now, just like when we talked about the other body systems, we would begin by talking about the system in general. Then we would go and talk about the cells and tissues that are part of that system. So when we did muscles, we did skeletal muscle and cartilage, right, and tendon. When we did the skin, we did the different types, the epithelia, plus the different connective tissues of the dermis. So here we're going to talk about the nervous tissue. Now, nervous tissue is basically one type of tissue. It's going to have neurons, which are the conducting cells. And then because those neurons are so specific, they need a lot of support. So then we're going to have a variety of different supportive cells called the neuroglial cells. And so here on this screen, you can see the neurons, the excitable nerve cells, and then the four main types of supporting cells. So the neurons basically conduct the electricity and the message and what we're going to call an action potential from one region of the body to another. Whether I touch, so let's say this little thing right here is sharp, so I pushed on that, I feel that, I feel sharpness. It goes up my arm, right, the afferent sensory towards the central nervous system. I feel ouch, I think I should stop doing that, and I pull my hand away. And that's because the brain sent a message to pull my hand away. That travels on these excitable neurons, the electrical signals, that electrochemical movement throughout the body. Then we have the four types of supporting cells, and here we go. So the astrocytes, and you'll see this guy is the white, and notice how it's clinging on to the neuron in yellow, different capillaries in red, and there's a lot of functions here. Look at them, 1, 2, 3, 4, 5, 6, 7, 7. Supporting, exchanging between capillaries and neurons, migrating of young neurons, controlling the chemical environment, influencing neuronal function. So these are the most abundant, the most versatile, highly branched, very connective, and they are really, really versatile, really, really, really important in the scheme of the nervous system itself. What I failed to point out, see what it says up there? The neuroglia of the central nervous system. When we think of the peripheral nervous system is out inside the body, right? We have nerves everywhere. They don't need these supporting cells. These supporting cells back here are central nervous system because the brain, right, all the cells and the neurons are packaged in the brain, and they are combined, controlled? What's the word I'm looking for? Sort of separate from the rest of the environment. And so these cells are found in the central nervous system only. All right, then we have what are called the microglial cells, the red one, again, touching the neurons. And they're monitoring neurons. They migrate towards injured neurons, and they can phagocytize microorganisms and other sort of dead neurons or dying neurons. So these are sort of like a defensive cell, sort of like a immune system function inside the central nervous system. Because again, the blood-brain barrier, the way that the brain is compartmentalized in the skull, it is separate in a lot of ways from the rest of the body tissues. Then we have these called ependymal cells, and the ependymal cells have cilia on them, and they move the central nervous system around, the cerebrospinal fluid around the brain, down around the spinal cord, up into the ventricles of the brain, and it bathes the cells in this fluid, and the cerebrospinal fluid is nourishing and moistening and protective, so these ependymal cells move that around. When we get to chapter 12, we'll do more with the cerebrospinal fluid and where it's located. Then we have these oligodendrocytes. Now, when we think about electricity, just in the wall, right? If I have a cord that plugged in, like this cord right here, is plugged into my computer. Now, I can grab onto this because it's covered in plastic. The plastic is not the conducting portion, right? Going from the wall to my computer. The conducting portion is the metal on the inside. If the plastic was not around that, and I grabbed the metal, I would get electrocuted. So the plastic is insulator. Just the way that this white myelin from the oligodendrocytes insulates the axons of the neurons. So the neurons are trying to send these chemical messages, send these electric conductions right down through the tissues. Well, in the central nervous system, we're going to cover those and insulate them with myelin by these oligodendrocytes. This next slide shows you the neuroglial cells of the peripheral nervous system. Now, there are some that are the same or similar, and others that don't exist or are not there at all. In the peripheral nervous system, the two major types of neuroglial cells are going to be what we call the satellite cells, which are those purple, looks like hair that's around the body of the neuron. And those are going to function like astrocytes. They're going to be protective. They're going to be functionally and structurally supportive, right, of the cell body. And then here we have the myelin in shorter segments, but still produced to insulate by what are called the Schwann cells. So the satellite and Schwann cells are peripheral nervous system. The oligodendrocytes, astrocytes, microglial cells, ependymal cells, and oligodendrocytes are central nervous system. All right, so a neuron. Well, this slide looks terrible. Neurons are the cells of the nervous system. They are conductive. So you'll see on the bottom picture, you'll see the cell body with all of these little branches coming out the top. Then you see a long, looks like a tail, a long process extending down to the right. So when we talk about the nervous system, we talk about the sensory input, the integration, and the motor output. And these impulses travel in one direction on one nerve. Now this is illustrating that. We have the neuron that conducts things in one direction. So it's going to come in through the dendrites and go out through the axon. So these are very special characteristics. They are, they last a long time. They're amitotic, which if you remember means they won't divide. They have a high metabolic rate, a lot of oxygen and sugar, and they have one or more processes. Here you'll see a neuron with 10, 20, 30 dendrites and one axon. You'll see the myelin sheath, you'll see the cell body, and notice the very bottom says impulse direction going in this direction. We would never see something going from the axon out the dendrites. It's always dendrite, cell body, and then axon. So the cell body is the center of the neuron. It's basically what we would consider sort of the cell. It's got the DNA, it's got the nucleus, it's got all different kinds of things. Now, if we sort of group those cell bodies together, in the central nervous system, they're called nuclei. And when we get into chapter 12, we'll see some basal nuclei and hypothalamic nuclei. Those are going to be clusters of neuron cell bodies. In the peripheral nervous system, arms, legs, etc., and chest, abdomen, we have ganglia. All right, and we'll look at some sympathetic chain ganglia and things like that in chapter 13. When we bundle neuron processes together, those are called tracts and nerves, and the processes can be dendrites or axons. So they sort of travel, sort of like thinking of a city. We have little streets, and they eventually feed a big main road, and that main road eventually goes to a highway, right? You can think of the main roads and the highways as the nerves and the tracts, and the little streets are going to be the dendrites and the axons sort of feeding together, but eventually they all fuse together, and that's what we're going to talk about, nerves and tracts. Again, we're going to talk more about that in chapter 12. So a dendrite, short process, cell bodies can have hundreds of these. They're receptive, they're the input region, right? And they're going to collect the sensory information. Right, this is a picture of a dendritic spine, a cell body, and you can see all of those feeding into that cell body. Then we have the axon. So the axon extends from the cell body. It's going to conduct the information away from the cell body. Some of the long actions are called nerve fibers, right? And then they all end at those axon terminals, if you remember from the muscle contraction. I mean, the axon terminal is sitting on the sarcolemma of the muscle fiber. Now we're talking about that axon. The axon is the conducting region. It generates nerve impulses and then ends at the axon terminals and then neurotransmitters. So we've sort of gone over this axon a little bit in the muscle contraction chapter. Again, myelin, it insulates the axon. It increases the speed of the nerve impulse. All right, so the more insulated an axon is, the faster that impulse is going to travel. So sometimes if there's myelination in the peripheral nervous system, then it's Schwann cells. If it's myelination in the central nervous system, then it is by the oligodendrocytes. And when we talk about the myelin in the central nervous system, we can actually see, which we'll see in lab, we'll actually see the difference between the white and the gray matter in the brain. And the white matter is going to be the tracts and the nerves and the axons and dendrites wrapped together. The gray matter is going to be mostly cell bodies and non-myelinated fibers. So we'll be able to see that physically when we dissect the brain as a part of chapter 12. Again, we have the dendrites, the receptive regions going towards the cell body. No myelin on the dendrites, only myelin on the axon, right, that sort of sends that impulse away from the cell body. So when we talk about neurons, these guys, this is a general structure of a motor neuron. There are actually 3 classifications of neurons based on the number of processes. So this guy would be a multipolar neuron. Has 1 axon and other number of dendrites, most common. We have also what's called a bipolar, 1 axon, 1 dendrite. Then we have a weird one called a unipolar. It has a T-like, it has 2 axons, which we don't see very often. All right, let's look at these pictures here. So here we have on the left, the multipolar. It's got the receptive region with the dendrites in the cell body. It's got that long axon, and then it's got the little secretory region, the little axon terminals. That's what we're used to seeing. In the middle, we have something called a bipolar neuron, and it's got 1 dendrite, basically, and 1 axon. Very quick, boom, boom. On the right-hand side, we have a unipolar neuron, which is really strange because we've been talking about 1 axon for this whole time. But for this one, we don't really have the receptive endings are really, really small, and it just goes straight through without even really going to the cell body. It's pretty interesting. So these are different pictures. This is, where we're going to find them. The multipolar neurons, we're going to find those most abundant, major neuron type in the central nervous system. So brain, spinal cord, predominantly going to be these multipolar neurons. The bipolar, very rare, right? Sensory organs, olfactory mucosa, which is smell, the eye and the ear. So very rare, we're going to find these bipolar neurons. And then the unipolar neurons found mainly in the peripheral nervous system, common in the dorsal root ganglia and some sensory ganglia of the cranial nerves. So again, we're really looking at multipolar neurons, but we wanted to talk about all 3 types. So we have multipolar, bipolar, and unipolar in terms of structure. Now we have functional differences. We have some neurons that are strictly sensory, the nerve impulse travels strictly sensory. We have motor, strictly motor. So, and then we have what's called an interneuron. They shuttle signals throughout the central nervous system. They're kind of an integration in the brain or in the spinal cord. So we'd have a sensory, purely sensory. Then we would have these little interneurons that will shuttle things around the brain. Then we'll have purely motor. So it's sensory, interneurons, purely motor. Most are within central nervous system, and most of them are interneurons. So when we look at sort of grouping those 2 things together, this is a really important table. So the multipolar neurons most of them are interneurons. Some of them are motor. The bipolar neurons, essentially all of them are sensory. And most unipolar neurons are also sensory. So multipolar, interneurons, and motor. Bipolar, sensory, unipolar, sensory. So read this one over a little bit more, just so you can understand bringing all of those 2 things together, the structure and the function of the different neuron types. Now, let's go and sort of change directions a little bit and think about the resting membrane potential, what we're going to call the resting membrane potential, because that is what we really want to talk about here. We really want to talk about the conduction of these cells. So here we have a picture of an axon, and you see the cell body and the dendrites, and it's looking at that little section. And notice how it's minus and plus, right? We're talking about electricity. It's not electricity like from the wall, but it's minus and plus. And minus and plus in the biological resting membrane is going to be the movement of sodium ions across the membrane. We'll get to that in a minute. But a resting membrane potential is approximately minus 70 millivolts, which means on the inside of the cell, it's negative in here compared to out here. All right? And so the membrane is polarized. It needs to be polarized because we need to have that change in order to flip it and cause right? Some sort of change to progress down the membrane. Just like we talked about when we did the action potential in the muscle cell. When the sodium goes out, right, and it causes that voltage change down the membrane, right? That's the same thing, voltage change, except now we're actually giving it a number. We're calling it minus 70 millivolts. So here we are. The extracellular fluid has a higher concentration of sodium. That's why it's positive on the outside. The intracellular fluid has more potassium. And we're going to talk about moving those ions across the membrane. Now, this would be a great video if I could play it. Can I? Strange. I wish that it would play and embed. I'm not really sure about how that works, but I would most definitely go watch the AP Flix resting membrane potential because that's going to give you a really good overview of it. So generating a membrane potential depends on two things: the differences in sodium and potassium concentrations and the differences in permeability of the plasma membrane. When we talked about permeability before, we talked about gated channels, voltage channels, right? How those ions are going to get across. All right, so here we have a higher concentration of sodium outside the cell, a higher concentration of potassium inside the cell, and you'll notice that we have some gated channels going to move those ions around. So the membrane is slightly permeable to sodium, it's very permeable to potassium, and a little bit permeable to chloride. And again, moving those around is going to change this positive and negative, right, potential along the membrane. So again, this is a really small picture, but you should try to read through it. The permeability of sodium and potassium across the membrane are different. In the three panels, we will build the resting membrane potential step by step. So potassium is lost through leakage channels. Sodium enters through a few leakage channels and reduces the negative membrane potential. And then sodium-potassium pump maintains the concentration gradient. We always have to end up at that negative inside, positive outside when we're talking about the membrane. Otherwise, we won't be able to send that signal down the axon. All right, so I'm just going to go through my notes here to make sure that I know where we're going. I don't want to miss anything. So when we want to talk about the different types of membrane potentials and how they change, what kind of signals, membrane potentials, again, change when the ions change, when the permeability change, exactly here, right? Where am I? Here. Differences in concentration and differences in permeability. That's how we're going to change our resting membrane potential. So what does that mean? When we talk about the action potential, we're going to start at minus 70 millivolts. And what we're going to do from there is we're going to decrease the membrane potential. We're going to move it from minus 70 up. That's called depolarization, right? And then we're going to cause a repolarization event and then what's called a hyperpolarization event, all by moving ions. Right? It goes from being less negative to being more negative, etc. We're going to look at that in a second. See here we have on the left, we're looking at this dendrite. The dendrite is more negative on the inside than it is on the outside. We are going to move that membrane potential towards zero. So we're going less negative, right? So we're going from minus 70 up to, looks to be about what, minus 60, something like that. That's a depolarization event. Here we have a hyperpolarization event where the resting potential goes down below minus 70. And what does this mean? On the left-hand side, we see the minus 70 go up to minus 65. What it means is inside that yellow, some of those are turning to plus, and we're changing that. We're lifting that negativity up a little bit. On the right-hand side, hyperpolarizing. We have the negative on the inside and the positive on the outside. We are actually contributing more negatives to the inside, reducing the negativity and bringing that down. What we're talking about on the right-hand side is a picture of the electronics of what's going on inside the neuron. So when we talk about these little graded potentials, they're little changes in membrane potential that are triggered by a stimulus that are going to open those gated ion channels. It's either going to increase or decrease the polarization inside the cell. And now we're going to go through these this process. So action potentials, right? Or we can either have graded potentials or we can have action potentials. The graded potential is going to be, short-lived, local, little tiny postsynaptic potentials. What we want to really focus on is the action potential, which we will pick up with on video 11B.